Okay, this kingdom here may start the same process that it may be split because you have ciliates, you have flagellates, and then you have the amoeboids, and then you have that other fungi like kingdom. World of it. So we know because of that breakdown that we know that the protists are a paraphyletic group, meaning that they have more than one common ancestor that gave rise to them. And that you see on the diagram up at the top right, that there's more than, it's not one arm of this of the image that gave rise to them, it's multiple. So we know that they had more than one common ancestor, meaning that there was a time period or a break in evolution that allowed that to occur. But we do study protists because we know that they can be harmful to us as humans, to crops, okay? And there are some good things we get out of them. So it's not like they're only a negative group. Okay. We know that they are eukaryotic. So we know that they have true organelles. So now we're moving from that prokaryotic world to the eukaryotic world. And we'll talk about the protists and the fungi, and then we'll move into plants down the road and eventually into animals. Because we know that the protista gave rise to all of them. Now, yesterday in lab, you would have seen the protists that you would have seen, most of them are going to be in the unicellular form. And then there were some algae that were multicellular that you should have seen, some green algae, red and brown algae. So there's a good variety of these. Protists are kind of a unique group of these, for the most part, single-celled organisms, as I said. Some can be heterotrophic, some can be autotrophic, and some can be both. Some are asexual, some are sexual, and some are both. So they have a large combination, which is why they are grouped into this category of this, unknown. So anything that was kind of similar, we just grouped together. The characteristics that we go by for the kingdom are as follows. Mostly unicellular. Notice that we say mostly, not all, because of the algae. They can be free-living or act as a parasite. Aerobic is the main form. True eukaryotes. Sexual and asexual, as we said. And they come in 3 flavors. Animal-like, fungal-like, and plant-like. The animal-like in that they're heterotrophic and they move. Fungal-like in which they use spores. And plant-like in which they are autotrophic. So that gives you a good sense of, like a broad sense of the kingdom. And as you said, it can be heterotrophic or autotrophic. So in lab, what you used to do was we just give you pond water, and you would just basically work through the pond water and classify the protists that you'd come across. And what you see there are the other samples. Like those are some of the things that you would find in that pond water. But now, since the lab changed and now that we're virtual, everything is done slightly differently. So now we're starting to move from the 2 prokaryotic kingdoms into this eukaryotic domain. Okay, so remember we talked about domains, okay? And each of these domains do only have 1 kingdom in it. And then you have the eukaryota, which will have the 4 kingdoms in it. And we know that Protista gave rise to the other 3, so we know that they fall into this category. So some of the things that make them very interesting to want to study is that they have a wide variety of habitats. Freshwater, saltwater, brackish water, which is defined as that in between fresh and salt. Inside of organisms, open ocean, on land. So they consume wide habitats. So it's not like they're isolated in one area. So it does make it a little challenging as far as saying, oh, you can only find them here. They're very diverse in that sense. So now we have to go to the reason of why we study them. And obviously the number one thing is so we understand them better, but there will be more to understanding them and how does that help us as humans. So as with everything, if it's going to harm us in some way, we have to understand how. So economically, if a protista are able to harm us, such as they're able to destroy crops, that's one thing. So your potato famine, if you were to grab this this potato at a store as though you were shopping for them, and you would squeeze it, you'd feel very soft, feel like a very bruised up banana. And because of that famine, economic loss was harsh at that time period. So we had to start to study them to figure out how are they getting to this into the soil, what's their life cycle, because not every produce has a straight life cycle, meaning I go from soil to the plant. Some protista have a life cycle that involves them to have 2 or 3 hosts to complete their life cycle. So we started to understand more about how these organisms work and understand that when you are an organism that has multiple hosts, that means you have multiple environments, which means that enzymatically things have to be changing. So we had to study them, and we understood a lot about enzymatic use here and also about the organism itself and its life cycle. Things can harm us. Okay, protista, a good chunk of them are going to be your everyday produce. So we do worry about those. The map here shows malaria, and these are the drug-resistant cases. Okay, so this is the cases that we have already messed up on by issuing the medication in the wrong methods. So obviously, malaria is something that we would be concerned about. This chart here, you don't have to memorize. I put it here just to give you a taste of some of the things that are out there. Some of them we talked about on Monday. In lab you would have seen Giardia, you would have seen Trypanosoma cruzi, the Trichomonas. Those are some of the things that you may have seen, but this chart isn't here for you to memorize. It's just here for you to see what's going on and the different things. So things like Entamoeba histolytica, it's an amoeba that lives, that once it gets inside of you will live in your small intestine and causes this thing called dysentery, which is bloody diarrhea. So it makes the infection so severe that your intestines are now leaking blood. So this gives you an idea of, there are many more, okay, of these protists that are harmful to us. These are the main ones that are found across the world. Just to go back for a second here, see your dinoflagellates are here. It is the next slide. They cause a thing called red tide, which accumulate inside of mussels and clams. And as you see here, this is that red tide. And what we have to understand is, in the image that you see here, we don't see what's to the right of that image. We just have that one screenshot of it, where we see the rock formation and we see that red tide. But these protists, like the bacteria, are not going to strive if there's no nutrients. What we don't see to the right of that is what was there that is causing some major runoff of some nutrients that allows them to reproduce and to create this toxin at that high level. Most likely there's probably a golf course or something present here where a lot of fertilizer is running off, causing this to have a good amount of the nutrients. So it's not uncommon that would happen. Okay. Now, because a good chunk of the protists live in the open ocean, a lot of research is going into how can we make those primary producers, those autotrophs that live in the open ocean, how can we make them better to help us? If we're able to genetically alter organisms by adding in plasmids into them, research is now looking at if we take these phytoplankton down here, and if we're able to have them reproduce at a higher rate and or go through photosynthesis at a higher rate, we're then able to fix more carbon from the atmosphere. Because if you think about it, the open ocean is 75% of the Earth's surface. So if we're able to make these reproduce higher and or do greater rates for photosynthesis, we're then able to fix more carbon and decrease the global warming effect that we have. In addition, because of that, now you're going to have more sugar present for all the organisms above. Because as the human population is growing, we already do overfish, but that overfishing obviously gets worse and worse as time goes on. So it provides the opportunity then to have more sugar. So we are looking at the protists, which are the base of the ocean food chains, as a way to help the environment and also help the food that is present in the ocean for us. If you look at phytoplankton, which is in the bottom right, and zooplankton on the top left. You would see how they look. We call it collectively plankton, but you can separate it into the animal-like ones and the protist and the plant-like ones. Plant-like are able to go through photosynthesis. Animal-like ones don't. It just has a general sense. And this is the diagram that's showing that, one, the absorption of carbon dioxide from the atmosphere to help with global warming, and the addition of food into the open ocean to increase food, increase the fish ability to reproduce, to want to reproduce more. Because a healthy, well-fed fish will reproduce at a higher rate than one that is unhealthy. So it's like a dual plus for us if we're able to figure out how to modify the phytoplankton to either increase photosynthesis or reproduce at a higher rate. So that's something that we're trying to understand and study. So we know that we have been trying to spend a lot of time on it. We know that we are working with them, and we're trying to understand their actual DNA sequence. So that's what we use to separate the monarans, the archaea, and the eubacteria. We're now in the same process of looking at their DNA for the protists to see if we can tell them apart. Because normally what we do is we just by how they look. If you're a ciliate, you get grouped together. If you're a flagellate, you get grouped together. But when you're dealing with a paraphyletic group, and that's a group that has more than one descendant, okay, that you have more than one common ancestor, it's not the best way to say, oh, you just all go there, okay? It's kind of like if you're rolling up change to return to the bank, you can't mix the different coins. They're all coins, but they all have different values. And when you mix them, you don't get the sense of how much you really have. Protists are the same way. You can't just assume because they're all protists that they're all the same. And that's what we did for years, and we're starting to really understand that that wasn't the best thing to do. So as you start looking at these protists, you can see that they do have some different branches going on here, okay, as far as what they can do and can't do. You can see how the connection between the protists and plants here, protists and fungi, and protists and animals. Okay, so this gives you an idea of how we know, or how we came to the conclusion that Protista gave rise to the other three kingdoms, to the plants, fungi, and animals. So one of the things that we had to explain is, how do you go from a prokaryotic organism to a eukaryotic organism? You don't just magically, move a wand and get that. So we had to have some explanation. So we came up with the endosymbiosis theory, just so we can define that a symbiosis is when two individuals of different species live in physical contact, meaning that they're in touch somewhere. And then endo meaning is that one organism is living inside of another. So our theory is saying that two organisms are living together, one inside of the other. So now we have to explain how you go from a prokaryote to a eukaryote under those assumptions. So using the image that you see here, you have an aerobic bacterium getting engulfed, okay? And eventually that bacterium is going to get the membrane around it, and start to live on its own, and eventually start to make energy. We feel that the mitochondria was a bacterium that was engulfed, membraned, and then started to function on its own inside of the cell to provide that cell with energy. So you have a cell able to engulf another cell, and then for them to live in theory, or in harmony, I should say. So this is the supporting data that we have. Mitochondria and chloroplasts are about the same size of an average bacterium. So we know that we have photosynthetic bacteria, we know that we have animal-like bacteria. Both organelles replicate by fission, as do bacteria, and have their own ribosomes, as do bacteria, and they manufacture their own proteins. Both organelles have double membranes, which is accurate with being engulfed. And the other thing, and most important thing, is that mitochondria and chloroplasts have genes that code for enzymes. okay, for their own genome, meaning that they have their own genetic information. So inside of us, you have a nucleus with your genetic information, and then you have a mitochondria with its own genetic information. And when we look at if we map out, just looking at the nucleotide sequence, you can see that chloroplasts, okay, which is an organelle, is very similar to the cyanobacteria, okay, when you're looking at the nucleotide sequence. And mitochondria and proteobacteria are very similar. So we draw those conclusions. Now remember that we're talking about a theory, just like evolution. Those are theories, things that we have evidence of, but we can't say are happening. Okay, that is 100%. Like gravity, that's a law. You can prove gravity, but we can't prove evolution because we have too many missing pieces that we don't have. But based upon the evidence I gave you, that's how we we feel you went from a prokaryotic world to the eukaryotic world. And there you had some major breakthroughs. So there's a sample of, you're looking at, you have Spirogyra here on the bottom left. You probably saw it yesterday in lab, an amoeba, okay? So you have a plant-like one, an animal-like one, and then you have your fungal-like ones. Now in lab, we generally don't look at the fungal-like ones. Tonight, we're briefly going to touch upon them because it's not a major topic for us, but we will mention the two phyla that are present for them. No, sorry, went too quick there. So plant-like protists, we know that they're autotrophic. I'm not going to go through photosynthesis, the light reaction and dark reaction for you. I would assume that most of you know the equation of how many carbon dioxide and water are present in the presence of sun to get you sugar and oxygen. And we also know that those autotrophic ones are going to be consumed by the heterotrophic ones to keep that food chain moving. So when we started to study them, we came up with protozoa, which means little animal, okay? These are named after these tiny organisms that moved, and we didn't have any clear understanding of them. And you can see I'm showing a variety of them, like a euglena, plant-like, in that it's autotrophic, but it also can be heterotrophic, but it's able to move. So I'm showing you here organisms that are able to provide some type of movement, like the Bordetella. You either looked at that or Stentor yesterday as an organism that itself can't move, but can make the environment move. By moving its cilia here, it can create that movement. So in looking at protists, some of them are very, very small, and some of them are large. Okay? Like your brown and green algae are very easy to see with a naked eye. But most of them are going to be close to a little bit smaller than a red blood cell. So what we're going to do now is we're just going to travel through the major phyla. I will provide you the phylum name. It might have been something you did yesterday in lab. And just talk about the basic characteristics of that phylum, just so that we're on the same page. So the first group are your Rhizopoda. Okay, these are your amoebas. They come either in a test or out of a test. When we say that it has a shell-like covering, they basically use the salts that are present and make a siliceous shell. Don't think it's something that's very firm and rigid. It's just something that will provide it next to barrier. But the characteristic of amoeba is that they kind of ooze along or move slowly through these things called pseudopods, these extensions. Okay? So basically what they do is they use the movement of liquid by changing concentration, forcing liquid to move into this area, swelling it, causing it then to extend outward. Once it's able to extend outward, that's how it's going to move, by using the mass movement. So really they're moving by changing concentration and keeping contained within the structure here, which we call your pseudopods. The pseudopods are used for movement and to engulf food. The food particles that you see here, these brown specks, those are, they just basically clean up the dead matter that's around. They don't go capture food because they just move too slow. But you probably saw an amoeba yesterday. Okay, here's the artistic drawing of it. So you can see the pseudopods coming out and the mass body back. Next group are your flagellates, okay? And their phylum is called the Zoomastogenea. Okay? The Zoomastogenea is a collective group of anything with a flagella. So the two samples I have here, you have Trypanosoma cruzi, which is an organism, it's a parasite that you contract through a kissing bug. And it obviously wreaks a little havoc on the face, causing, I guess, swelling would be the best way. To this guy down here, which is a flagellate that lives in the gut of termites. And it lives in the gut of termites. It's able to break down wood, but the termite itself can't break down the wood. It's the actual flagellate that's doing it. So I just grouped these together because they both contain the flagella, whether it's a single flagella, as in a Trypanosoma cruzi, to multiflagellated that you see here with the organism at the bottom. And they would be classified into your phylum Zoomastogenea. The ciliates, anything with cilia that will create movement. You can put Stentor in here and Vorticella that we had just looked at earlier. Those can be organisms that you could drop into this category as well. Okay, that would fall here. So anything with cilia, we drop into what we call these ciliates. And the last of the protozoans are these things called the sporozoa. These are ones that are parasitic, they're non-motile, but they are animal-like in which they are heterotrophic. Okay, so they are carried, in this case, you can see that the organism, okay, the protist, must be carried by the mosquito and the human to complete its life cycle. So this one has two hosts to complete its life cycle. Okay, and you can see there's no mobile structure here that is found. There's no cilia, there's no flagella, and there's no pseudopod. So now we'll move into the plant-like ones. Okay, and when you're looking at this type of a pond here, where it's just covered in green, understand that the algae on top is blocking all the sunlight from going down. So anything underneath there is not going to do well. And in general, when you see something like this, it would go along with what we had talked about earlier, that there probably is some runoff of some fertilizer here that's allowing this to be a very nutrient-rich zone in which the algae are going to strive. But understand that the plant life underneath is now blocked. So it is a handicap, you know, but obviously the good thing is that it's making a lot of oxygen for us. There are some perks to it. So we have three unicellular grouping for the algae. You have the Euglenophyta, the Basilorophyta, and then your dinoflagellates. Okay. The Euglenophyta, it's a common one, you probably saw it yesterday. It's green, it has the eye spot. It has a flagella that's just not shown here, but it's probably going down into the screen itself as we're seeing it. This is your Euglena. Okay, it's heterotrophic as well. So it is plant-like. We group into the plant-like category because it can go through photosynthesis, but understand that it also is animal-like that it's able to move and also can be heterotrophic. So it goes both ways in that sense. The Basilorophyta, these are your diatoms. Sometimes we use them in the lab, sometimes we don't. But they're characterized by having a shell around them made out of salt. And it's such a thick layer of the salt that as the light passes through, it creates different colors. It creates like light prisms. So that's why you're seeing them in different colors. It's based upon how the light is going through them. The different shapes go along with the different species. But the diatoms are probably one of the largest oxygen makers in the world. Okay, and that they live in the open ocean. So that just gives you a sense of what they would look like. They come in different shapes, sizes, and colors. And these are your dinoflagellates that you saw earlier, creating here, you see like your red tide. You have the research vessel out there with a line trying to capture data for it. But these are your dinoflagellates. They are able to go through photosynthesis, but also are able to move. Okay, so they're plant-like and animal-like. Okay. The difference on them is that they are not heterotrophic. Where the Euglena can be heterotrophic and autotrophic. These guys are autotrophic only. So when the red tide does happen, it doesn't create a major kill-off, as you saw earlier, as you saw Monday with the anoxic zone. That's slightly different. Okay, but this is what you would see. It will kill off some of the organisms because it's a toxin. So now we'll move into the multicellular. We'll cover green, red, and brown algae. Okay. Red algae comes in different shades of red. Don't always assume it's going to be like a nice brilliant red or like a pinkish color, because you have the one up here which has some darkness to it, but it has the red hues to it. So all the multicellular, all these are going to be multicellular because they have the red hue in them. We grouped them into the Rhodophyta. So this is phylum Rhodophyta for these for this group. The brown algae characterized, the main one is kelp that create these air bladders. And understand this is a great evolutionary adaptation for the brown algae in that if you create air bladders, and the air bladders, as you would understand, would want to rise to the surface, it takes that kelp and brings it higher up on the water column because the higher up it goes, the greater the light intensity, and therefore it'll be able to go through photosynthesis at a higher rate. So if it was to stay across, if it was going to hold fast at the bottom of the ocean and not be able to elevate itself through the water column, it's not going to get as much light. So the idea of creating these air bladders is pretty original, okay, and different. Green algae, most of you have seen as far as walking through streams and lakes and ponds. We group it into the Chlorophyta. There are, I forget how many different species of them, but they're in all these. Just so I can go back for a second, there are multiple species. It's not just one species. As you saw here in the red algae, I showed you 4 different. There are many more than 4. Okay, this is just for the means for the class that we're just talking about them as a group, because the idea is you understand collectively who they are. If this was to be your purpose of study, then you would study them further and be able to go and separate within the phylum what are all the classes that are present and what's the difference between the classes. This course is not about that. It's a broad survey. Here's looking at Volvox. You might have looked at that yesterday in lab, where it's a colony of cells that are living together. You can stain them whatever color, but they generally are a greenish hue because they will go through photosynthesis. But in lab, when we look at them, they're normally pink. So you might have seen it as pink. I don't want you to be confused, but they normally will have a green hue to them. Condomonas, okay. Another plant-like with a flagella. So basically, I'm showing you all this to give you an idea of how diverse this kingdom is. Even though it's unicellular and small amount multicellular, there's a lot of diversity. And we talked about the amoeboids, the flagellates, the ciliates, and we gave a sample basically of each, but there are many samples inside of there, which is a lot of people feel that in the next 20 years, the protist kingdom may be divided into 2 or 3 other kingdoms based on what the genetic analysis comes up with. And then our last grouping that we have for the protists are your fungal-like ones, where you have they live as your decomposers, and in one case, in the Mymaxicata, they're actually able to create movement, and they do it the same way we had spoke about with the amoeba by changing concentration inside them. They're able to move water, and as you know, when you move water to one side, it creates a heavier mass. They then start to create a movement or an oozing. So that's why we call them slime molds, because they actually kind of move like slime. They move very slowly and move across. But there are 2 known phyla of protists that are fungal-like. Okay, so we do know that there are 2. So any questions on the protists?
Nope.
Okay. So what we'll do is let's take a 5-minute break. I have 8:40 on my clock. Let's come back at 8:45, and then we'll go through fungi, which will probably take us a half an hour, maybe 35 minutes to get through. Okay. So I'll take attendance when we come back. Let's take a short break. The main thing. Okay. Not necessarily the whole part of it. So, and just looking back, just to go through some of the stuff a little bit that we had covered, I didn't record just so we can just go through it. The first thing that we did was we just looked at how they live. Okay, they live as decomposers, they live as parasites. Okay, and then lastly we talked about them living as in this mutualistic relationship. So we know that fungi are made up of hyphae that are able to grow. Most live as decomposers, as this one here, showing you it's breaking down dead matter in the soil. And the reason why they get so large is that if you look where my mouse is here, as they eat the dead matter here, unless if something comes along and dies there, there's no more food. So they constantly are expanding. I believe the largest known fungi was over 1 square mile, and that was in the rainforest of Ecuador, if I remember correctly. That was the largest one ever documented. Okay. Now, because of their enzymes, as we said earlier, that they're able to digest, then ingest, which is the direct opposite of us, where we ingest our food, then we digest our food. That is important. So they're heterotrophic by absorption. You are heterotrophic by ingestion. Much different. The next part that we went into, just to recap for you, is the three ways in which they are going to get their energy: decomposers, parasites, and then living with an algae to get sugar. This is over 90% of the fungi live in this state, okay, as a true decomposer. Very few live as a parasite. I showed earlier athlete's foot. This is showing you shelf fungi, which lives on the plant. And again, just like the hyphae of the athlete's foot going into the animal, this is the hyphae of the shelf fungi going into the plant to steal nutrients. And keep in mind that a parasite is defined as an organism that harms another one to gain benefit. And then the last way in which they live to get energy is as a mutualist. Okay, living with algae, where they break down dead matter and they give those nutrients to algae, kind of like a fertilizer per se, and the algae in exchange is able then to make sugar. All right, so then we went into the different phyla, which is where we conclude our conversation, where the first phylum are the Chytridiomycota. Okay, that's the first grouping, where they have that motile spore. The Ascomycota are characterized by a thing called an asci. And if you look at this, you notice that they have these spores going in a linear pattern, and they're in 8s. And that's the characteristic of the phylum Ascomycota, is that whether they are in a structure like this, where it kind of looks like a broom or hair-like structure, or a cup, their sexual structure is called an ascus because it has 8 spores. And if we look at a drawing of it, what you wind up having is a zygote that's able to go through meiosis and then mitosis. Now, one would think for ease, it would probably be better to go from one and then to go through mitosis to 2, and then from 2 to 4. Okay, or sorry, I mean 2 to 8. So the reason why we feel that the organisms did this is that there's more variation when you go through meiosis. Remember, mitosis, you're just making a copy of yourself, right? It's an identical copy. But in meiosis, you have things like crossing over and the assortment of the alleles that leads to variation. So by the ascus evolving to create the ascospores by going through meiosis first, they increase variation. Not dramatically, but every little bit helps. Okay, so this is what you would see as far as, just to go back to here, and you'll see in lab, you'll see the Ascus having 8 spores in them. Okay, they come in different stuff, such as truffles and yeast. Okay, there's more than one type. In lab, this is what it would look like if you look at it in lab, showing the Ascus. Okay, these individual lines that you're looking at are what you would consider like the gills of the mushroom, the underneath part. Zygomycota. These are the major food killers for us. Okay, and they create a thing called a zygospore. Just looking at as far as bread mold, in lab, sometimes they grow bread mold for us and you're able to look at it. And we look at bread mold up close underneath your dissecting microscope. My always assumption was that the mold was growing, the black stuff that we saw on the mold was, on the bread was directly on the bread, but it's actually elevated up. Those black spores are high up. And this is, if you had a piece of bread like this, I would recommend you not eat it. But if you had just a corner of it, you can break that off. But understand that when you break off that, or when you see mold on bread, there are so many spores, there's already millions of spores already present there, that you're not going to, that loaf of bread, if it's still a whole loaf, is not going to last much longer. Okay? Because as you move the bread, all these spores come off. Any movement will cause spores to be released, and you can't see them with the naked eye. So they land on the other pieces, and as you know, bread is nothing more than sugar. It's a great place for them to grow. And you see what happens is the spore will germinate, creating the hyphae. The hyphae eat through. So when you actually see the bread mold itself, this stage, remember the mushroom is already grown. All the mushroom is in the mass. You don't see all the hyphae. So when you break off this corner and eat the rest, you actually are eating a lot of the hyphae, which won't hurt you. So the question from Serenity is: so wait, if you break off the molded part, you're still eating fungi? Yes, because the hyphae are still in the bread. She probably doesn't want to talk about that anymore. But it's not going to harm you. We're eating spores all the time. The hyphae in there are not going to hurt you. It's just like eating mushroom. It's no big deal. Okay, but just understand that when the bread mold got to the point that you see the mold, it has already grown inside of the bread. Okay, it's already there. Muhammad writes, even if it's a lot, yeah. Actually, just so you know, Muhammad, that once you see the spores present, the blackness of it, it has already grown a lot inside of the bread. So keep in mind that hyphae have grown through the bread and probably grown from one slice to the other slice. The mold is a reproductive structure. So it's already gone to that adult stage, using air quotes, that it's already developed enough to get to that stage. So it's already, you're going to get a lot out of it. Okay. So the zygospores are thick-walled, and some of them have structures on them. You see here, these are elaters, we call them. They're like wings. So when the wind blows, it takes it further away and spreads it out. Those of you with kids, you would know that if you have kids, I have 2, that having a kid growing right next to you, taking up the resources, it's difficult. As an organism where you can't move, if your offspring is growing up right next to you, that means that you have to compete now for the resources. So evolving these things called elaters or wings, some can float, they create flotation devices, so they can float away. There's a variety of different things that they use, but what the whole goal is to get the spore away from themselves so that they're able to germinate somewhere else. But that's the characteristic of a zygospore. The last phylum that you look at in lab, okay, so you have the Ascomycota, the Zygomycota, and then this one, your Basidiomycota. These are characterized by your shelf fungi or your bracket fungi, rusts and smuts, which are devastating to crops. And keep in mind that once they're at this stage, the number of spores that it produces are so hard to regulate. So it's not an easy thing to manage once it gets to that level. Just if most of you know what a mushroom anatomy looks like, so you have the cap, and if you turn the cap over, you have all these little lines called gills. Inside of those gills are where the spores are, and this is what you're looking at. You're looking at the spores in between the gills. So that's what you're seeing here. These are your spores inside, in between those gills. So that airspace in between the gills, that's where the spores are lined. And as wind circulates through there, it blows them off and brings them with them. Then our last grouping are the imperfect fungi. We don't know their sexual structure, which is why we don't have a lot of information on them. Fungi are characterized by a sexual structure, but now we have this new fungi that we know is present. It's genetically different, but we haven't been able to isolate the sexual structure. So that's the last phylum that we have. It's not one you look at in lab because we don't have the sexual structure to really work with for the most part. And last but not least, this is the reasons why fungi have been such an important part of understanding science or just biology. Okay? The need for nutrients, genetics, okay, your central dogma idea, growth, development, and also differentiation. So many different things we have learned by studying mushroom because they germinate easily, you get next generation quickly, and they are a eukaryotic organism. So it allows us, and they're closer to the animals than are the plants, as we saw by their genes. So we've concluded for today. Are there any questions you guys have?
Professor, I looked in the email. There was no code. The invite email?
So when you log into Test Wizard, what are you seeing?
There's two options, grades and class, I believe.
Okay. So when I set up an exam for you, it will appear there for you underneath your class. Okay. Who am I talking to right now? Mohammed. Okay. You should see it as a class. Once the exam appears, you'll see it pop up.
Okay.
You are set up and able to run, so you should be able to be fine with that.
All right. Thank you.
Not a problem. So just to recap everything for us, we covered protists and fungi today. You will have a